In this video tutorial, we demonstrate how to work with BugSigDB data in R using the BugSigDB R bioconductor package. We will build on the contents that we introduced in the tutorial on basic usage of BugSigDB. And we recommend that users that wish to work with BugSigDB data in R familiarize themselves with these contents first. The installation of the BugSigDB R package has two prerequisites. We first need a recent version of R, and second, we need the BioC Manager package as the recommended tool for installing bioconductor packages. R can be downloaded from the comprehensive R archive network under gran.r-project.org. To install the BioC Manager package, we proceed by carrying out the following command in the R console, which will first check whether the BioC Manager package is already installed, and if not, we'll proceed with installing the BioC Manager package. Once we have BioC Manager installed, we can proceed with installing the BugSigDB R package using the install command from the BioC Manager package. The goal of the BugSigDB R package is to facilitate import of BugSigDB data into R and bioconductor. We also note that the BugSigDB R package is primarily a data package. Users interested in descriptive statistics and comprehensive analysis of BugSigDB contents are recommended to visit the BugSigDB stats package, which continuously integrates with BugSigDB.org and provides weekly updated analysis on study, experiment, and signature level. The first step after installation is to load the package, which we can achieve here via executing the command library of BugSigDB-R. Once loaded and attached to our current R session, we can start making use of BugSigDB-R's functionality. For example, the import BugSigDB function, which allows to import the complete collection of curated signatures from BugSigDB. The dataset is downloaded once and subsequently cached, providing for fast access to the dataset and avoiding redundant downloads. It is also possible to use the argument cache equals false to force a fresh download of BugSigDB and overwrite the local copy in your cache. We can then proceed with inspecting the resulting data object, which is a standard R data frame with each of the more than 2,000 rows corresponding to a microbial signature from differential abundance analysis. In addition to the signatures themselves, the resulting data frame is richly annotated with additional metadata columns, providing information on study design, antibiotics exclusion, sample size, experimental platform, and statistical analysis, among many others. Subsetting the full dataset to certain conditions, body sites, or other metadata columns of interest can then be done along the usual lines for subsetting data frames in R. For example, the following subset command restricts the dataset to signatures from microbiome studies on obesity based on fecal samples from participants in the United States. The resulting data frame then contains 34 out of more than 2,200 signatures that are satisfying the defined subset criteria. Given the full BugSigDB collection of signatures, or a dedicated subset of interest, we can then use the function getSignatures to obtain microbial taxa annotated to each signature. The resulting object is a standard R list that contains an entry for each of the 2,270 signatures. If we look at the first three signatures, we see that each signature consists of a descriptive name and the microbial taxa annotated to that signature. The signature name can be decomposed into an identifier string that lists study, experiment, and signature ID, followed by the study condition and the contrasted sample groups. Here comparing conventional adenoma cases against control samples. The last part of the signature name indicates whether the annotated taxa were found with increased abundance or decreased abundance in the study group. The microbial taxa annotated to a signature are returned based on NCBI taxonomy identifiers per default. However, this is an argument to the function and can be changed using the tax ID type argument that is NCBI per default, but could also be the full taxonomic classification in metaflan format or the taxonomic name only. Two fields of particular importance are the condition and body site fields, which are standardized based on the experimental factor ontology for condition and the Uber anatomy ontology for body site. This provides for leveraging ontological relationships between conditions or body sites and for higher level queries against BugSigDB. The BugSigDB package provides the getOntology function to import both ontologies into R. For example, we can use the function to here import the experimental factor ontology, EFO. The resulting object is an object of class ontology from the ontology index package, describing here relationships between roughly 35,000 experimental factors. As we have demonstrated before, we can obtain subsets of BugSigDB signatures for certain experimental factors or specific body sites of interest. Similarly, we can subset the full BugSigDB data frame using higher level queries that involve ontological relationships with the subset by ontology function. This facilitates grouping of signatures that semantically belong together. For example, we can use the subset by ontology function here to subset the BugSigDB data frame by the condition column to all signatures associated with different cancer types. More specifically, this subset will restrict the condition column to all descendants of the term cancer in the experimental factor ontology that are present in the condition column. Carrying out this ontological subset command, we arrive at a data frame that now contains 305 signatures associated with different types of cancer. We can inspect the various cancer types that are present in BugSigDB, for which signatures of differential microbial abundance are available by using the table command. The resulting table lists the number of signatures that are associated with different types of cancer in BugSigDB.